Thank you. Thanks for the invitation to come back this year. Let's see, get this going. I have no disclosures to make. The objectives for this presentation will be to describe some of the pilot studies of genome sequencing that are going on in newborns, to talk about or understand some of the counseling issues that come up with families whose infants have had genome sequencing, and to list 3 ethical issues associated with genome sequencing of newborns. So with newborn screening, we think about the heel stick that you heard about earlier, and also the screening for critical congenital heart disease through pulse oximetry, and screening for hearing loss with audiometry studies. What is newborn screening? In a task force that published their findings in Pediatrics in 2000. I like their definition. It's a public health program aimed at the early identification of conditions for which early and timely intervention can prevent or reduce associated mortality and morbidity. There are criteria for effective newborn screening. These were taken from screening criteria that Wilson and Youngner published in 1968. And these include that treatment is available, that early institution of treatment before symptoms manifest reduces or eliminates the severity of illness, and that otherwise you're not going to detect this in a newborn, right? They're going to look fine. As you heard well from the family earlier, that their baby looked perfectly normal. They had no idea until they got that phone call that there could be anything going on. And it's the same for physicians when we see newborns in the newborn nursery with various conditions that we are able to screen for and do screen for. They otherwise look fine. And also an important part of effective newborn screening is that there be a rapid and economical laboratory test that's highly sensitive, so ideally no false negatives, and reasonably specific, few false positives. Because there was a lot of variability from one state to the other in terms of what conditions were being screened for across the country, in 2005, HRSA funded a task force that was through a contract to the American College of Medical Genetics, and this group came up with a recommended core panel of 29 conditions that they said all states should screen for, and also 29 additional secondary conditions that would be detected as part of the screening for the core conditions. This group, last I knew, is now called the Discretionary Advisory Committee on Heritable Disorders in Newborns and Children, and they continue to meet on a regular basis. New conditions can be nominated. And I think, unlike with the earlier group, they go through a very detailed process of looking at the evidence for various conditions. So it's very evidence-based, which in the past, they did the best they could, but I think it's become more of a formal process in looking at, how common is this condition, what's the data about it, if we do screening, are we going to be able to detect it, and so on, is there treatment available? People, groups, a lot of parent support groups are behind some of the nominees for conditions that the advisory committee considers. And some of the limitations for not including recommended or conditions that have been considered were that there was no treatment available, there was not a good screening test for that condition, or the screening tool was too expensive, and so on. And so if you look at the initial group of conditions that people considered, it was There were, the ones that made the cut, which would include PKU. Actually, the number one was MCAD, which was one of the main ones that was included in the expanded newborn screening that first started in the late 1990s, early 2000s. That was through tandem mass spectrometry when that became commonly used in state newborn screening laboratories. But then there were a lot of other conditions that they considered that sort of didn't make the cut. And if you look at the total numbers and percentage of those that were excluded, 23 out of the 30, so about a third, were excluded. And the reason for them being excluded was that there was no test available, right? You wouldn't pick them up with tandem mass spec or other types of analysis. 21 of these 23 conditions that were excluded are detectable in some or all cases with molecular genetic analysis. So if we talk about doing next generation sequencing, whether it's whole exome sequencing or whole genome sequencing, you could really overcome a lot of these barriers in order to diagnose these conditions. So if a genetic etiology for that condition has been established, theoretically, we're able to detect it with genomic testing, as you heard earlier from Dr. Brower. So you'll also sometimes hear this described as massively parallel sequencing. And what I like to use as an example for our students is, if we think about traditional sequencing in the laboratory where those letters are looked at, the nucleotide base pairs, the GCAT, it's sort of like you could, with a single test, you would be able to read what's on, a letter in that mailbox, but and that was called Sanger sequencing, the traditional types of individual sequencing of genes. But with a whole exome or next generation sequencing, it's almost like having the entire encyclopedia in your mailbox and being able to read on each page what is present. So theoretically, next-gen sequencing can search for mutations in all genes, all 20,000 known human genes. As also you heard from Dr. Brower earlier, whole exome refers to just looking at the coding parts of genes, the exons, where we know the majority, although not all deleterious variations are present, or as Dr. Kingsmore in Kansas City, with his rapid turnaround time, is doing whole genome sequencing, which would include both the exons and the introns. But as you've also heard, our analysis is very complex because our understanding of what's a significant variant or mutation and what's just a polymorphism has a very long way to go. As another brief overview of next generation sequencing, what the pipeline is, it's really fairly straightforward. It's no different than what we do for other types of genetic testing. You have your patient, you collect a sample of DNA from them, you prepare that DNA, you do a library preparation depending on what you want to look at, and then the actual sequencing equipment is very small. It fits on the top of a laboratory bench. But then, as you've also heard, the informatics portion, all the computer portion is very critical for this and challenging, takes a lot of computer support and bioinformatics support. You go through an aligning, you go through looking at your raw data, your VCF files, and then trying to figure out, separate out what are the polymorphisms from what are the significant variants. And I like to point out this part because it's really this validation by humans that have to look at the information and figure out what's significant and what isn't. And that's really one of the most time-consuming parts of this. where, Dr. Kingsmore in Kansas City is able to do it in, about 3 days or so. For our studies, for our research studies, it generally takes us about 6 to 12 months or so to get all the information together. Now, we're not doing clinical whole exome. I know, for example, at Baylor, they have a much more rapid turnaround time for their clinical exomes that they do. But as a research study, it does take many months because we don't really have the people that can devote full time to looking at the information. But it's not just what comes up as possible significant variation in the gene, but we have to go to the literature, to PubMed, look at articles, a lot of things that have been published that, this, mutation, this is a mutation, and it may have only been found in one family. And so the data really doesn't always support it. So you have to be very, very careful before you think of this as being a significant change, and especially if you're going to be returning results back to patients or families, and also offering to put it in the medical record. You have to, be very, very cautious. So when we think about a change in a gene, just made up of hundreds, thousands, sometimes millions of these base pairs, and a variant in that gene may just be one single change from a C to a T, and that's what a gene variant is. So there are about 3.8 million of these variants in every one of us. And again, the vast majority are not deleterious, don't cause any problems. Most are just polymorphisms. The definition of a polymorphism is that variant is present in 1% or more of the population. Most are benign, but some are not benign. They're pathogenic because they adversely affect how, for example, the gene functions in terms of making a protein. Or if the gene has a regulatory process that it goes through. And so, again, just figuring that out is a challenge. And we find these changes that we really don't know what the significance is, and we utilize our variant calling software. There are a number of different ones. Some are homebrewed, some are commercially available. We also use databases, the 1000 Genome Project, where they take populations and look at what variants they've had in supposedly normal, healthy individuals. We utilize ClinVar, other things like dbSNP. Exact is a newer one that's available that I think has a million or so subjects that they've looked at. So it's getting better, but it still has a ways to go. Also, if you have the time and financial means to do modeling, you can put that mutation into a model organism, one commonly used as a fish called the zebrafish, and you can look and see what effects that variant has on the zebrafish. Needless to say, although it's a fairly rapid way of doing it, it still requires quite a bit of resources. What are some of the limitations of whole exome sequencing? I think people falsely assume that, you can just detect everything if you're going to do a whole exome or a whole genome. And we know that right now it's not very good at detecting these trinucleotide repeat disorders, such as Huntington's disease and Fragile X syndrome, for example. And I think for pediatricians, that's very important to keep in mind because I've actually seen a couple of kids now who have gone through very fancy genetic testing. They've had, maybe karyotype, normal microarray, and then somebody's jumped to a whole exome in them. And they have not tested for Fragile X syndrome in a child with developmental delay and intellectual disability. I haven't had any yet that came back positive for Fragile X, but it someone would look quite silly if they spent $10,000 on a whole exome and didn't do Fragile X testing that was just a few $100s. So I think that's an important thing for people to keep in mind. We also know that certain parts of our genome are not very well analyzed through whole exome, and that would include areas where there's a lot of the nucleotides GC. So GC-rich areas are difficult to analyze. Also, again, some deleterious changes are present in the introns, other areas of the genome that wouldn't be picked up if you just look at the exons. Copy number variant conditions such as DeGeorge syndrome, Williams syndrome, and so on are not very easy to detect, although that is getting better. And then structural variants, like a couple, let's say, who's had 3 or more miscarriages, who would be at risk for one of the members of that couple having a chromosome rearrangement, a balanced rearrangement. That's not something that you would want to do whole exome for, because you wouldn't pick up balanced rearrangements like that, although again, that's improving. And then some of the epigenetic changes. There are certain conditions where there's no deletion or duplication in the genome. There's no change in the nucleotide base pair, such as some forms of Angelman syndrome and Prader-Willi syndrome, where it's due to an epigenetic change, which typically affects methylation of the gene, so it has to do with gene silencing, and that's not something that you would pick up with an exome. And also you have to be aware of pseudogenes, so other regions in the genome that have the same gene sequence as the gene of interest, but it's not a functioning gene like the one you're interested in, and you can often mistakenly pick those up and find changes that really, aren't significant. So you have to be aware of that. For the majority of us, still the turnaround time is an issue. It does take quite a while, as I said, to get the results back, and cost is still a major factor, with on average for a clinical genome is around $10,000, of which most or many insurance companies aren't covering it, and at least in our state, our Medicaid program doesn't cover it. They don't cover microarrays either, so they're pretty limited in what they do cover. Pretty much no genetic testing do they cover. And another problem for autosomal recessive genes or disorders is that you can't tell, if you find 2 variants, you can't tell whether they're on the same chromosome, which would be in cis, or are they on the 2 separate alleles or 2 separate chromosomes in trans, in which case then it's likely that there is a significance, whereas if that genetic change is on the same allele, then it's unlikely to be causing the problem if it's an autosomal recessive condition. So still a lot of limitations. On the ethical side of things, there's a lot of issues, of course, that have come up with utilizing this. These incidental or secondary findings. So testing can identify variants that are not at all related to the original intent of the testing. And for next-gen sequencing, these can include mutations in genes for adult onset conditions like breast cancer, colon cancer, cardiac arrhythmias, and even childhood onset conditions like intellectual disability. So, we've all thought a lot about, what should be reported back to patients and families. Other things that come up, well, I told you about the criteria for newborn screening, that these should be treatable conditions. So if we're going to be thinking about using whole exome or genome sequencing in newborns, and can detect some of these other conditions, but are these conditions really treatable? So one reason I got involved in this area was a project that we had that looked at doing newborn screening for Fragile X syndrome. So a condition associated with developmental disabilities, and typically children with Fragile X are not diagnosed until they're 3 years of age, even though parents often bring up some concerns earlier on that their children are not doing quite right. But we still don't have an effective treatment for Fragile X in terms of a medication. There are various trials that have gone on that are, using different medications to see if there might be some success. So far, none has been found. But then again, those of us who take care of children with disabilities, we know that early intervention is very helpful. Just like we refer a child with Down syndrome for early intervention services, we're not going to cure the Down syndrome, but we're going to help maximize that child's abilities. And it's the same for Fragile X, and it's the same for most conditions that are associated with developmental problems. So shouldn't we be broadening our definition of what's treatable? So also we know that for families who have a child with a problem, oftentimes it does involve their child's development, but it could be other things, other kinds of medical problems, and they go on what's been termed this diagnostic odyssey, going from specialist to specialist, having, MRIs and CTs and maybe muscle biopsies and various single gene tests and metabolic studies. And if you could zero in more quickly on what the cause of that problem is, isn't that a benefit? Also, you could avoid unnecessary and expensive testing, invasive types of testing. And you could also provide the family with genetic risk information. For Fragile X syndrome, oftentimes families have had 2 or more children with Fragile X before the first one is diagnosed. And then if you think about some of these additional conditions that you might detect that might not really be going to impact the child during childhood, but maybe later onset, but is benefit of testing, does it only have to be for the child, or could it be benefit for the other family members? And for example, with a BRCA mutation, the breast cancer genes are inherited in an autosomal dominant pattern. And so if a child is found to have it, odds are that one of the parents also has it. So if you are able to detect that in a parent and you save the life of that parent because of that information, you get them screened, and isn't that benefit to the child if you save the life of that parent? And then on the other side of things, those involved in pediatric ethical issues argue that, well, if you're going to test a child and give information about adult onset conditions, you've taken away that child's right not to know that information, right? So that you've taken away the autonomy of that child to be able to decide when they're old enough to understand and think about it, whether or not they even want that information. But then we also have to balance the parents' rights to know information about their children. And I think there's no right or wrong. I mean, everybody's got different opinions about this. Also, not just for adult onset conditions, but for carrier status. If we find that a child carries a gene for cystic fibrosis or PKU, where we know they're not going to be affected by the condition, and they really don't need to know about it until they're adults and thinking about having children of their own, but yet that would mean that probably they got that gene from one of the parents, and maybe the other one's also a carrier, and maybe they're at risk for having a child with a recessive condition. There's still concerns about, will all this cause genetic discrimination, right? That when that child grows up, that because of this information, they won't be able to get various types of insurance. We know that we do have GINA, the Genetic Information Non-Discrimination Law. The act was passed and signed into law in 2008, and I figured I should show this guy's picture because President Bush was president when it was signed into law. It took effect, began to take effect in 2009, and it's to prevent discrimination in health insurance and employment-based situations for genetic information. So to prevent your employer from utilizing genetic information and either not hiring you or firing you from your job, and having insurance companies that provide your health insurance charge you more or deny you insurance because of the knowledge about genetic information. But it doesn't include members of the military. So military folks are not protected through GINA. And it also doesn't include life insurance, disability insurance, long-term care insurance. So only right now our medical insurance. So with that as a bit of a background, I'm going to talk more specifically about a project that we're involved in, and then briefly 3 other centers that receive funding from NIH. So in 2012, NIH put out a request for applications to explore the implications, challenges, and opportunities associated with the possible use of genomic sequence information in the newborn period. And 4 centers were funded under a U-19, which is sort of a large multi-investigator method, and the 4 groups together are called Insight. And I'm going to go through briefly with slides that the other centers shared. One of the centers is at Brigham and Women's Hospital and Boston Children's Hospital in Boston, and they're actually looking at 2 different groups of children. One would be a group of healthy newborns, 240 healthy newborns, and then 240 from their NICUs up there. Both groups will be randomized into 2 separate groups. So one will just get standard newborn screening and a family history will be taken. The other group will get those 2 things as well as having their genomes looked at. And then the same with the group in the NICU. And they will get genetic counseling before and after. And they do plan to place the information into the medical record and share it with other healthcare providers. They'll study these children and their families up to 10 months after they have the genome sequencing done. And they'll be able to provide the primary care providers with information that may not be something to be concerned about right away, but if that child were to develop problems later on, like developmental delay, they'll be able to look at that information for clues about what might be going on. The Children's Mercy Hospital group is really just focusing on the critically ill newborns in their NICU, and with their rapid turnaround time and looking at the turnaround time is really the emphasis for their project. And they'll have one group that will be randomized and will just get like standard types of genetic testing, like if the clinicians think that a child might have a genetic mutation that's associated with like neonatal seizures, for example, they may send out just a panel to look for those genes, and then they'll be doing a whole exome on a similar cohort of patients. And they'll follow up long term. In California, at University of California, San Francisco project, which is also working with the newborn screening group in California, they're taking a bunch of stored anonymized blood spots where they know what the diagnosis was, and they're looking at whether they can pick up on whole exome sequencing the mutations that would be associated with those conditions. They also have a subset of patients who have severe combined immunodeficiencies, and those where there's not been a specific diagnosis that has been found, and they're looking at whether whole exome can be helpful in providing a specific diagnosis for the immunodeficiency problem. And then our study, we call it NC Nexus for North Carolina Newborn Exome Sequencing for Universal Screening, funded by both NHGRI and NICHD. And our overarching aims are to evaluate whether next-gen sequencing can extend the utility of current newborn screening. Can we devise and evaluate a clinically oriented framework for doing what we call next-gen sequencing newborn screening, and develop best practices for incorporating next-gen sequencing newborn screening into clinical care. In order to get funding, you had to have 3 projects that were all sort of equal strength. And project 1 involves generating and analyzing whole exome sequence data sets and generating a large amount of information by doing this. So some of the analytical strategies, how to do your variant calling, how to communicate with other computer systems about different things. So really the whole bioinformatics part of it is important. Let's see, if we look at the filtering that you have to do, when we're getting results on a whole exome, we're not getting bombarded with 100,000 different results. Our computers are able to get it down to in the order of a few hundred variants that then they give a sort of a letter grade to, depending on what they think is likely to be significant, is a VUS, or is likely to be non-significant. And although there's no gold standard, some of the things that we're working on are ways to better determine what is significant and what isn't. What we get out is through our workflow manager, and this just shows us whether this is a VUS, a likely benign variant, or so on. But again, as I mentioned earlier, there's still a lot of people hours that have to go into this. Project 2, which is the part that I'm most closely related to, is the clinical part of it. One of my interests has always been in genetic causes of hearing loss. And so one thing we're doing is to look at whether whole exome sequencing can pick up some of the conditions that we are detecting through standard newborn screening. What's the sensitivity of it? So we're also looking at the specificity of it. For example, a child that we know has PKU, are we going to be able to pick up mutations in the phenylalanine hydroxylase gene that causes PKU through our whole exome sequencing? Also, for example, if we have a child with hearing loss, a baby's diagnosed with hearing loss, congenital hearing loss, picked up through newborn screening, they go to audiology and have that hearing loss confirmed, but parents have no idea what's the cause of the hearing loss. They get on the internet, they Google, they see things like Usher syndrome and other syndromic forms of hearing loss and wonder, is my baby not only going to be deaf, but also go blind? Things like that. And right now, that's not part of standard newborn screening to figure out what is the cause of the hearing loss. That's recommended that child who's got confirmed hearing loss go through genetic testing to try to figure it out, but that can often be, a very expensive, lengthy process, not available to all individuals. And so those are the things that we're looking at, as well as sort of the ethical issues which overlap with our Project 3. And we, for various reasons, we're not going to use blood spots. We're using saliva samples for a number of reasons, but a little bit easier to get through a research study. And parents certainly don't seem to mind when we just ask to swab the baby's mouth. And we're getting very high quality DNA from that saliva. So our project 3 is really the ELSI issues. So when the Human Genome Project was started, a portion of the budget was put aside to study the ethical, legal, and social issues involved in the Genome Project and Genomics, and that has continued with NHGRI continuing to fund groups that look at what we refer to as the ELSI issues. So one of the things that we're spending a lot of time on is to categorize genes into various bins, and I'll go into that more in a minute. We're also, as part of our informed consent process, because it is so complicated to try to explain to parents what's a whole genome, what results might you get, do you want your child to participate in this study or not? So we're developing, with the help of our partners at RTI International, a nonprofit company that's close to us in Research Triangle Park in North Carolina, but they've got expertise in these decision aids for parents. So we're developing an online decision aid that will help provide the information to families about this. And anyway, so going on to how we're thinking about different genes. So, 20,000 genes, it's really hard to go through and think about each one. There are certain ones that we know are associated with early onset conditions. And so my colleague, and I want to point out Dr. Jonathan Berg, who's my co-PI on this big project. He's involved in a lot of other studies, including the ClinVar studies. But he and Jim Evans at UNC came up with this scoring method to help us sort of think about genes. We've modified it a bit to be more significant for the pediatric population. But basically it's looking at 5 different areas, and it includes the severity of that condition. What's the nature of the threat to the health for an individual who carries a deleterious allele in this gene or a mutation, okay? Is it, this level 3 would be sudden death or inevitable death in childhood. Number 2 would be possible death due to disease or severe intellectual impairment. Or is it a 1, serious morbidity or moderate intellectual impairment, or 0, modest or no morbidity? And then what's the likelihood if a person or child has that gene variant? What's the likelihood that they'll develop the condition? Also, term that you can use for that is the penetrance. Is it pretty high, greater than 50%, or down to less than 1%? And then, are there interventions, and what's the efficacy of intervention? Is it highly, are we really able to, reverse the problem or highly treat it, or is it, moderate, minimal, ineffective? And then also, what's the acceptability? How acceptable are the interventions in terms of burdens or risks placed on the individual? Is it being on a special diet, or is it, going in for a colonoscopy? Excuse me. Or is it, really something like, having one's stomach removed because you find out that you're at risk for a gastric cancer, for example. And then what's our knowledge base? For a lot of things, there's only 1 or 2 papers out there. For other things, there are hundreds of papers, there's guidelines, there's gene reviews, there's things like that. So the total score could range from 0 to 15, and that's what we term our medical actionability score. So if you find mutations in this gene, how important, how significant is that? An example for PKU, so the severity of the disease, it is associated with severe intellectual disability. So it got a 2. Likelihood of severe outcome, it got a 3. The effectiveness of the intervention with diet got a 3. we know, as we've heard about earlier, very effective if you're on the diet. It got a 2. It's not the easiest thing to be on the special diet, but it's not one of the worst things, and there's a good knowledge base for it. So then a condition that I thought, well, you know, isn't this one that if we had a method of screening for, and it's a cancer condition that can definitely affect children. It's multiple endocrine neoplasia type 2B, and it's associated with early childhood medullary thyroid carcinoma. Usually by the time it's diagnosed, children have metastatic disease already. There's 100% penetrance, so if you have a mutation, you're going to develop it. And then also they can have pheochromocytomas. About 50% of patients with it will have pheos, and then other things like mucosal neuromas. There is a sort of a Marfan syndrome-like body habitus, but it's usually not very evident in childhood. The prevention is pretty straightforward. You take out the thyroid, and then later on to screen for pheos, you do checking for catecholamines. 50% of cases are de novo, so don't expect to find a family history necessarily. The prevalence for all forms of MEN2 is 1 in 35,000, so fairly rare, but not that different from a lot of conditions that we screen for nowadays in newborns. The gene is known. It's called the RET gene, and there's good genotype-phenotype correlation. This is an older boy, 2 boys with MEN2B, but again, you know, the features really wouldn't be noticeable in infants. And I just show this to say that there are certain mutations that give a higher risk. So there is what we call genotype-phenotype correlation. So if you find, you know, certain mutations with a high risk, the age at which prophylactic surgery to remove the thyroid is recommended is in the first year of life. For some of the others, the onset of thyroid carcinoma is a little bit later, so you can delay that. And there's no population screening method currently available for this. So if we go back to our actionability score and we look at MEN2B, so severity of disease, possible death due to cancer, got a 2. Likelihood of a severe outcome, fully penetrant, got a 3. Effectiveness of interventions, remove the thyroid, prevent cancer, got a 3. The acceptability, yes, you have to go through a surgical procedure, but it's not, you know, extremely difficult to go through. So that got a 2 in our scoring system. And then there's good information in the literature about it. So it got a 3. So that actually got the same score as for phenylalanine hydroxylase. Another example, familial adenomatous polyposis, colon cancer condition. Again, severity, possible death due to cancer, highly penetrant. You start doing colonoscopies early, and I should mention that children with FAP gene, APC gene mutations can also develop liver tumors. So we also screen them for development of liver tumors, and that's actually earlier in childhood compared to the onset of polyps. So acceptability, specifically just thinking about colonoscopy, it got a 2. Again, a high knowledge base, so that one also got a 13. If you look at a condition like Tay-Sachs disease, hexosaminidase A is the gene associated with that. Again, there's inevitable death in childhood, highly penetrant, but right now there's no effective intervention, although there are research studies going on to look at various treatment possibilities. And so the acceptability of intervention, since there's no intervention, that got a 0, a high knowledge base, but that got a score of 9. And what we call our binning committee is a group of individuals who are clinical geneticists, clinical biochemical geneticists, genetic counselors, maternal fetal medicine geneticists, bioethicists, social medicine people, and our metabolic dietitians also participate in our binning process. So it's not just, one or two people who are coming up with this scoring. We each are assigned various genes and discuss them in a large group setting every 2 weeks. You can also kind of modify things depending on when the age of onset is. And so if you look at the upper left-hand quadrant, which would include genes like for PKU and MCAD, RET, we are going to include these in what we call our next generation screening newborn, or next generation sequencing newborn screening. And then the other ones, such as the adult onset ones that are medically actionable, like BRCA1 and 2, would be in the upper right-hand quadrant. And then there are those with onset in childhood for which there's no medical actionability, like Tay-Sachs, Rett syndrome. Duchenne muscular dystrophy is in that group, although there or some benefit to early onset of steroid treatment in boys with Duchenne. And then there's the group of adult onset non-medically actionable, such as early onset Alzheimer's and so on. So our NGS, NBS would be those conditions that are currently in the recommended uniform screening panel and those that fit a similar profile to the RUSP conditions, utilizing our algorithm. And then we're using 2 different cohorts of patients, one with diagnosed conditions and the other a healthy newborn cohort. For those with diagnosed conditions, they will get back known pathogenic variants and variants of uncertain significance in the genes that are associated with their known condition. And then both groups will get the NGS, NBS results, but we'll have a very high level of calling something a pathogenic variant. So we don't want to report back VUSs in these because, we, especially, for the healthy cohort who are shouldn't, really have any underlying conditions, so this is going to be very rare. But if we find, let's say, a RET gene variant that looks like it's pathogenic, we would return those results. And then the families will be randomized into 2 groups in each area. One will only get the NGS, NBS results. The other will go through a process of using the decision aid with trying to decide what additional, if any, categories they want to get back, such as the adult onset medically actionable conditions, carrier status of the child, and those conditions like Fragile X syndrome and other conditions like Angelman syndrome, for example, associated with intellectual disability, but for which there's no traditional type of treatment. And for those up here in the this group, adult onset, non-medically actionable, we would not report those results back, so no one would have the option of learning about, risk of Alzheimer's disease, for example. The decision aid is one that RTI is developing, again, online, and we hope that this will be something that could be utilized on a broader scale and available to other places if this goes forward. And they've also helped us with this discrete choice experiment where we're looking at how do parents decide about, what information they want to get back from this. And this was something that I wasn't familiar with before, but it's basically giving parents multiple choices. So it's not just this or that. It's kind of like, well, if there was a condition, for example, where 50 children out of 100 with this gene change will develop the health condition, and the age of onset is young, and it's associated with severe mental disability and mild or moderate physical disability, would you want that information versus another condition where only 10% of children would develop it at an older age with mild to moderate impairment, and just getting this. And they had over 1,000 families that they surveyed about this. asking them, which profile would be more important for you, and there are a lot of different variants in this. And the bottom line was that the likelihood of occurrence was really the main thing that people were concerned about. So they were much more interested in finding out about conditions where there was, a 90% or more likelihood that the child would be affected versus, a low chance. So, and they're continuing to analyze some of the results of this, and I hope to have more in the future. And we gave them actual disorders, to think about, like things that would impair vision versus things that would impair intellectual development, and looking at that. So anyway, it's a complicated study. This is sort of the procedure that people will go through from initial recruitment, all these various times when they'll be surveyed about their thoughts. They'll look at how they go through this decision aid, and we have a lot of different folks involved in it. So can next-gen sequencing expand the utility of newborn screening? It certainly will allow us to test for additional conditions. It will improve the specificity and sensitivity of standard screening. Already, molecular testing is utilized at the newborn screening level, either as a first or second tier method, or when confirmatory testing is done when children get to the medical centers. For CF, it's part of most standard screening protocols. It's utilized for some of the hemoglobinopathies. For SCID, it's utilized generally after the newborn screening lab has found the condition, and then when they go in for confirmatory testing to figure out, what's the next step. Oftentimes, molecular testing will be used to narrow down what's the specific cause of the immunodeficiency. People are looking at, for PKU, are there genotype-phenotype correlations where the specific mutation might dictate how well a patient, responds to Kuvan or PEG-PAL, some of the other treatments that are being looked at. And certainly, as I mentioned, like for hearing loss, it's very helpful to zero in on what the cause of the hearing loss is. And this is just utilizing looking at the recommended uniform screening panel and the different genes that we know of that are involved with the various disorders. I don't think it'll ever be a standalone test, but I think that there will be targeted next-gen sequencing panels used that zero in on these conditions that are most important to detect in newborns. People are utilizing integrated screening models, so both traditional types of newborn screening along with next-gen screening panels. And still, we have the ethical questions. If we don't return information to families, where should it be stored? How do we store it? Whose responsibility is it? What about genes that we may not know of any clinical significance now, but a month from now, maybe we'll learn that it is something clinically significant? What are parents' rights to their child's DNA sequence? How do we go back and try to find families? It's already hard enough when we, get information from standard newborn screening sometimes to track down the families. So if you're talking about, several years down the road, how are you going to do that? And some of the commercial labs are already offering next-gen panels to supplement newborn screening. They're not, marketing this directly to the public, but to physicians, pediatricians, geneticists, and so on. Should things be mandatory or just voluntary? Should they only be utilized by people who have the financial means? But then what about health disparities if we're going to do it that way? And just the demands on the public health and healthcare systems that are already overburdened with things. And then the whole issue of genetic discrimination, I think, is something that we have to certainly not forget about. So this was something that came out through MIT online review a few months ago, but it was a graduate student at University of California who was able to get a hold of some DNA from his unborn child and thought, oh, hey, it's cool, let me do whole exome sequencing on it, and said that got these results and got 7,000 different, variants. And afterwards, he said sequencing his son in utero was more cool than practical. He did it to show where technology is headed and because he likes to, quote, push the envelope. And said that our attitude is that you make a lot of decisions for your kids, including ones that may seem sketchy in hindsight. So I think he wasn't too happy that he had done that. But anyway, so the future is now, I think. And I'd like to acknowledge again my co-PI and brilliant scientist Jonathan Berg, others at RTI, Don Bailey, Megan Lewis. Myra Roche is our genetic counselor on the team. Chris Reaney is in the School of Public Health. Laura Milko is our really everything person for the project. Kirk Wilhelmsen heads up the bioinformatics team, and many others who are involved in the binning committee and other parts of the study. So I was trying to find something funny, but there's not a lot of things that I think are very funny in cartoons, but at least about genetics. But so this little girl says, I just got my 23andMe data, more than 500,000 genotypes. Her friend says, cool beans, what does it all mean? And they're like, well, we don't really know. But and the little duck says, so many genes, so little time. Thanks. I'd be glad to answer any questions.
So, okay, there's a lot of information, and I think I may have not followed the entire talk. My question is, so, the diseases we are screening now, I think in most states, we are all, most of us agreed they're very relevant. And I think one concern is the more you find out from, the entire genome, it may not be relevant. So remind me, with your next-gen newborn screen, will you, when you do that screen, how many disorders right now which you screen do have a genetic basis, where we know? You know what I mean? So,
yeah, so I think there probably won't be more than like 100 that would fall into that category. So I don't think it's going to be thousands of different things because they really don't meet the criteria that we're looking for, right? So it'll be fairly limited. It's part of the whole project is to figure out which ones fall into that category.
And this study, has it actually started, or is it going to be?
Well, so we're not actually sequencing anybody yet. I didn't bring this up, but we've run into the FDA. And the FDA has become very interested in all of these projects. The FDA actually contacted us. While some of the centers have changed their protocols to satisfy the FDA and not have to go through a big rigmarole of all the required documents and stuff, but we've sort of stuck with our original plan. And the FDA is very concerned with the fact that we're going to be screening children for adult onset conditions. Now, for a lot of conditions, you don't really know when the onset will be, childhood or adulthood. But anyway, they've gotten very interested in it and are requiring us to put in what's called an investigational device exemption, and which is taking a huge amount of time and effort that we hadn't planned on. So we're a little late. We'd hoped to start screening in the spring, but it probably won't be until later this year.
Okay. Because one of my concerns is, we screen so many children, and most of the screens which, where we repeat the screens or we call the parents to finally come back normal. Mm-hmm. So I guess at least one way of going about would be to screen, just take the present screen, but whatever we can do genetic screening on those, if there's a way to do those rather than doing, hundreds of diseases, make all those phone calls, the anxiety, probably that will be reduced.
I know. Yeah, we're hoping to survey pediatricians on their thoughts. I mean, I can understand it's hard enough for people to deal with the standard screen. And again, remember, these are research projects. This is not anything that we're advocating for prime time anytime in the future. It's far too expensive to think that a newborn screening lab is going to start doing whole exome sequencing. But the idea is that there are reasons why it can be very helpful to provide more specificity in the test results that are obtained. And, ultimately, we may start doing this. I think the real benefit is going to be more on that critically ill newborn group, where you're trying to figure out, what's the cause of their problem. So,
Thank you.
yeah.
So
Yes.
the AAP has recommended, among other things, a chromosome microarray for developmental delay.
Yes.
I'm not sure what the difference between that is and the whole exome.
That's a great question. So the microarrays, remember, pick up copy number variants. So they pick up fairly large chunks of DNA that are either missing or extra. So they'll pick up things like DeGeorge syndrome with a deletion of, I don't know, 3 million base pairs of nucleotides. The whole exome sequencing picks up a single base pair mutation or small missing or extra pieces of DNA. But as I said, it's not good for those big chunks. It's not good for DeGeorge syndrome, really. And so the copy number variants are the ones that are, in 15-20% of patients, the cause of their intellectual disability. I think ultimately it'll be the next step. I mean, that's why, Baylor and others offer clinical whole exome for these kids that they look like they have something genetic. They've got severe intellectual disability. Their karyotypes are normal. Their Fragile X is normal. Their microarrays are normal. And then it's been the next step for, trying to figure out what the underlying cause is. So it's a higher degree of being able to look at the genome is this whole exome next generation sequencing. The microarrays are sort of in between, but they're a very strong tool for certain types of conditions.